Signalomics in Regulatory Sciences.
Thank you very much again for that introduction. We will be looking at Signalomics in regulatory sciences. Before starting on this topic, which is a topic of where we are developing a significant amount of research in looking at signals, we would look at the background to this. And the background surrounds the movement to move towards a patient-centered ethos. A patient-centered ethos surrounding the three pillars on which we base regulation, namely quality, safety, and efficacy. And to these pillars, we add accessibility and the environment. Our competence is to look at the tools that we have available in regulatory sciences. And these are some of the established tools that we look at: good manufacturing practices, we look at quality control, quality assurance, and also to more recent aspects of regulation in regulating newer areas such as quality by design. Against this background of regulation, we took the idea of looking at the need to quantify and validate signals. Everybody is used to see signals. You could see traffic lights as signals. You could see signals as a perception to take action. And what we are thinking is of taking prioritization and assigning a value to ascertain the value of signals. The idea is to develop signals from a perception into a science through the establishment of a robust, evidence-based signal management system. Now, we have undergone a number of research in this area, such as that by Benjamin Micallef in determining the signals that we had related to the COVID vaccine, any signals for toxicity, in an environment where signals were of extreme importance because there wasn't the time in developing the vaccines in going to the normal evidence-based methods. And what usually would have taken 5 years of research was done in a few months. The decision-making relied a lot on signals. Unfortunately, we don't have the science that signals were still treated as a perception. And that is what probably produced the vaccination hesitancies in some places. Another study that was carried out was a very good study by a person who has experience in the field in quality control, and this was the study by Adriana Spiteri that looked at the signals in a quality control and which of those signals have actually led to a real occurrence. And so a way how you could give a basis, a number to those signals that would produce those signals as evidence-based. Another research project that was carried out by Paula Cardona-Schwireb, and in this she looked at incidents related with 107 medical devices. Could the signals in those during the incident, before the incident, were there signals before the incident occurred that if taken through a scientific approach, one would have avoided the occurrence of this incident through taking action, regulatory action, on the signal and not on the occurrence. So taking regulatory action, which could be drastic, on signal management. There were other areas like the environmental impact in drug development, where there are signals that could have predicted a serious impact and evaluate the impact on that area. And finally, we are having at present a PhD student who is working very closer to home on the evaluation of and seeing whether we could identify signals and make those signals evidence-based, and thus using signals for quality assurance in medical products regulatory sciences. Today we also had the good news of having another PhD student who would be working on signals related to medical devices. We are presenting this new science of signalomics, and we are thinking that we could prove that this is a new frontier for innovation, where it is surrounded around a data-centric innovation strategy that we use this data as a proactive strategy to enhance public health by fostering advancements and promoting scientific rigor through signal identification and management. Thank you very much. Anyone interested in this area may contact us because we are looking for collaboration in this newly developing science. Thank you very much.